Hello everyone, this is Chandrasekhar from Chandu Biology Classes. In this particular session, I am going to explain to all of you the entire 2023 TSSET question paper. Okay, so to be specific, all the 100 questions which was asked in the TSSET question paper, all the 100 questions were appear from Chandu Biology Classes notes only. I will explain every question very briefly and very elaboratively. I am requesting all the aspirants watch the entire session to get a good knowledge and to understand how to solve a question. Before that, so please find out the details about the upcoming batch, and especially these are all the achievers of TSSET 2022. I mean to say the exam which was happened in the 2023 March, right? And these are all the details about the new batch. The new batch is commencing from the 28th August, friends, and you will get the coaching till October 28th, before the day of the exam. So you will get all the live and recorded session. Entire entire TSSET life sciences paper 2 will be covered. You will be provided the study material. You will get all the chapter wise test, all the mock test, and you will get a 24/7 chat assistance also. So whomever are very sincere or very dedicated that you want to crack the exam, so please call or WhatsApp on this particular number for the further details. Okay, now let's start the session. So look into the first question. Actually, friends, I can say that 2023 TSSET life sciences question paper is the most easy question paper so far that we have seen. If you look into the 2020 2019, that time the question paper was very tough, but this time the question paper was very easy. But we don't know how the question paper will be in the upcoming days. But every student has to read from very basic to the very advanced level, then only you will be in a position to crack any kind of a question. Okay, now look into the question number 1. In the process of the reverse transcription, it's actually an intermediate level. What is the reverse transcription process? Every student commonly know that this is the central dogma from where from the DNA we will synthesize the RNA, which we call it as the transcription process. Anyway, you all people commonly know that later RNA get translated into what, friends? A protein. That also we know it very well. We also know very well 90% of all the organisms are having DNA as their genetic material. You know it very well. That is the reason from the DNA, most of the organisms synthesize the RNA. Finally, RNA is going to give rise to the protein. But if you look into the organisms like a viruses, they do not have the DNA as their genetic material. Instead of the DNA, they're having RNA as their genetic material. Since they're having RNA as their genetic material, what happens? By using RNA, they are going to synthesize the DNA. So synthesis of RNA from the DNA, we can call transcription. You know what is the definition I can give you? DNA dependent RNA synthesis. We can call it transcription. And friends, but we are synthesizing the DNA from the RNA. So what we can say that RNA dependent DNA synthesis, which we can call it as the reverse transcription. There is a chance that they can ask the question in the exam about the enzymes. All the students keep the point in your mind. What is the name of the enzyme that is involved in the transcription? DNA dependent RNA polymerase enzyme. Every student commonly know that. But since we are talking about the reverse transcription, here the enzyme is RNA dependent. No need to get confused. This is very important. RNA dependent DNA polymerase. Why DNA polymerase? Because we are synthesizing the DNA. From where we are synthesizing the DNA? From the RNA. That's why we can say RNA dependent DNA polymerase. Anyway, to the given question, what is the reverse transcription? Where the DNA is getting synthesized from the mRNA. Majorly happens in the viruses. No need to get confused with that. So now, look into the second question. A very simple, straightforward question. Which one of the following amino acid has a pKa value near the physiological pH. I think most of the students we know very well. Actually, friends, what is the speciality of the histidine? Histidine, I hope you all people remember. What is the side chain of the histidine, friends? A histidine having a imidazole group. What is the pKa value of this particular imidazole, which is 6? So all the students look into this. Histidine having a pKa value of its R group, that is the imidazole ring, which is the 6. So I want all the students has to understand, since it is very close to the neutral pH, you know what we can say, that partly it can act like an acid, partly it can act like a base also. Please don't get confused with that. This is the only amino acid that can act as partly acid also, as well as a base also. What about the other amino acids? Either they are acidic in nature or basic in nature, but mostly histidine is basic. But the point is that, friends, even it can act like a acidic as well. So what's the amino acid having the pKa value near the physiological pH? Histidine. It's a straightforward question, right? Now look into this. Glucose reduces in amylose are linked by. Every student know that this is also intermediate level question only. Every student commonly know that starch is what, friends? Starch is a large polysaccharide. Every student commonly know about it. And you also know that if you break, starch is a polysaccharide. If you break down this particular starch, what do we get, friends? We get amylose, and we get amylopectin. And one point is very clear. The amylose and amylopectin, if you break down, what do you get? Undoubtedly, you will get the glucose only. So what I'm trying to explain you, glucose is the monomer for the starch. I mean to say, many glucoses come close together, they form amylose and amylopectin. Amylose and amylopectin are finally going to give rise to the starch. That we commonly know that. Now, what's the question they're asking? How does this amylose? So glucose also contain amylose only. Let's consider 200 to 2000 glucose units are present in the amylose. How they are getting connected? You can see the picture very clearly, friends. So first carbon of one glucose can be connected to the fourth carbon of another glucose. So what will be the linkage? The linkage will be the alpha 1,4. If you look into the amylopectin, it contain alpha 1,4 also, as well as, friends, it contain the alpha 1,6 also. There is no doubt about it. So what will be the answer now? Alpha 1,4. That is the answer B. Very easy, straightforward question. Next one. Glucose is converted to the mannose. See, what is glucose? Every student commonly know that it is a monosaccharide. You know it. What is mannose? You also know very well. Mannose is also a monosaccharide. Now the question is that they're asking, how does the glucose is getting converted into mannose? That is the question. Very simple question. All the students look into this. Glucose having how many carbons? You can see the picture. Glucose having 6 carbon. Mannose having how many carbons? Undoubtedly mannose also having a 6 carbon. What is happening? Look into the picture. So there will be a shifting of the position at a carbon number 2. If you look into the second carbon, OH group is on right side, hydrogen is on the left side. But you just see the mannose, OH group on the left side, hydrogen on the right side. This is what we can call shifting of the position. So this generally we can call epimerization. A simple, straightforward, direct question. Now, friends, if you look into the same, so this time they have asked about the mannose, next time they may ask about the galactose. So how does the glucose can be converted into galactose, friends? Again, epimerization at the carbon 4. So glucose converted to the mannose by the shifting position at the carbon 2. Glucose can be converted into galactose by shifting the position at carbon 4. No need to get confused with that particular point. Now, very important, straightforward question it is also. Vitamin D is derived from which of the following precursor by the action of the UV light? So first of all the students has to remember a common point. vitamin D is produced where, friends? You know very well vitamin D is produced in the skin. If the vitamin D want to be produced, friends, compulsory it require a precursor molecule. Precursor molecule means what, friends? Precursor molecule means starting material, from where this particular starting material is coming from. And what is that starting material? 7-dehydrocholesterol. Now please look into the diagram, all the students focus. So you can see this, right? So UVB, what light, friends? Ultraviolet rays B, it will activate 7-dehydrocholesterol present in the skin. Then the 7-dehydrocholesterol converted into the pre-vitamin D, and pre-vitamin D later modified into the vitamin D. A very simple, straightforward question for the exam, right? So answer will be the D. Now, what is the question? Which of the following amino acids are considered as glucogenic amino acid as well as the ketogenic amino acid? This is also directly from our notes, a very simple, straightforward question. So I'm requesting all the students to remember compulsory the amino acids which can act like a both glucogenic and ketogenic. It is very important, friends. You have to remember compulsory because there is a lot of information to be remembered. One is called isoleucine, another one is called as lysine, phenylalanine, tyrosine, and tryptophan. These amino acids can act like a both glucogenic and ketogenic. Very simple, right? What is the glucogenic amino acid which are involved in the production of glucose? Ultimately, glucose is getting converted into what? Pyruvate. Simply remember the amino acids which are going to give rise to the pyruvate. They are the glucogenic amino acid. And what are ketogenic amino acid? Ketogenic amino acids are those type of amino acid which are involved in the production of the ketone bodies. That is called ketogenic amino acid. So among given amino acid, which is the amino acid act like both glucogenic and ketogenic, friends? Undoubtedly tyrosine. There is no doubt. It's straightforward question. Right. Now, C-value paradox suggests that. So every BSc student commonly know that, and every MSc student also commonly know that it is a direct question. See, friends, the answer will be the D. First of all the students has to understand what is the C-value paradox. Every student commonly know that every single organism having their own content of the DNA, correct na? Which is constant for all the individuals. Yes or no? Every individual DNA is present. You also know one common point. Listen, every student. 99.9% of the all human beings having DNA which matches 99.9%. So the DNA can be differentiated from one person to the another person hardly 0.1% only. Correct na? So remember, so each species of organism has a characteristic content of DNA which is constant in all the individuals of that species. That is only we generally called as a C value. No need to get confused with that point. The amount or content of DNA of a species is not indicative of the number of genes. Yes or no? Of course, it is very difficult to explain based on the content of the DNA, friends. You can't explain the number of genes. That's why it is called C-value paradox, which means non-collinearity. I can say that there will be a no proper relation. Try to understand. There's a no proper relation between genome size and complexity of organism. See, what I'm trying to explain you take an example of a bacteria. Bacteria is a very smaller, but the number of genes present in the bacteria are more. So what I'm trying to explain you, genome size and the complexity of organism is entirely different. There will be a no relation. That is what explained by the C-value paradox. No need to get confused with that. Now, the question number 8. The genes of color blindness in man are located on. Every student commonly know about this particular point, friends. The gene for color blindness is actually located on which chromosomes? Undoubtedly on the X chromosome, which you have studied in the 12th standard also. It won't present on the Y chromosome. It won't present on both X and Y. Either X or Y also wrong statement. It is strictly present on the X chromosome. Always remember, in case of a color blindness, who will be affected more? Of course, friends, male affected more. Always remember that particular point. And what about the female, friends? You all people commonly know that female generally act like a carrier. Please don't get confused with that particular statement. I want all the students has to understand. A RNA polymerase holoenzyme consists of the following subunits. First of all the students has to understand what do you mean by the word holoenzyme. Every student try to understand RNA polymerase having a two enzymes. One is called core subunit, another one is called sigma subunit. Friends, core subunit, sigma subunit together, we called it as the holoenzyme. It is also straightforward question. What do you mean by the word holoenzyme? Holoenzyme means, friends, a complete enzyme. Every student commonly know that. So what does this particular core subunits are made up of? Core subunits are made up of alpha, alpha 2, beta, beta prime. So these are all core subunit. When I add the sigma factor, then core plus sigma becomes a holoenzyme. So the answer will be undoubtedly option C. I have explained it in the properly in diagram also. There are two units of alpha. You can see that one beta prime, one sigma. All together we can call it holoenzyme or a complete enzyme. No need to get confused with that point. Very straightforward question. This is also you read in the degree level itself. Balbiani rings are associated with. Every student commonly know that they are associated with a lamp brush chromosomes. Correct na? What is Balbiani? It is the name of the scientist who identified the ring-like structures in the lamp brush chromosomes. You all people commonly look into the picture. All the students look into this. Okay, so actually, friends, all these particular polytene chromosomes as well as the lamp brush chromosomes, they are actually very large chromosomes. I want you to understand that particular point. They are very, 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 very large chromosomes. And I want you to understand where this particular lamp brush chromosomes are identified. That is also very important for the exam, right? Where they are identified and why we have to remember that. Oh, I'm so sorry. I'm extremely sorry. I'm sorry. Balbiani rings are associated with what? Polytene chromosomes. I'm sorry. Right. So now the point is that try to understand my statements very correctly. If you look into the structure of a polytene, polytene means what? There are very large chromosomes which are having thousands and thousands of the DNA molecule. What, friends? They are having thousands and thousands of the DNA molecule. Now you please try to look into the picture. If you look into this, see this is the entire giant chromosome. The giant chromosome consists of different type of bands. It is having a dark bands which give a thick color, and it is called interband which will give a thin color. And most important, friends, so they contain a ringed structures. I can show you like this. You can see this, right? They will appear like this. Chromosome puffs. That chromosome puffs. Puffs means you can see this. Like this, chromosomes are present in a group. Now you can ask me a question, sir, what is the use of this Balbiani rings? I can tell to all the students very clearly, Balbiani ring is the region where the active transcription of RNA is taking place. What, friends? Always try to understand where there is a abundant amount of active transcription is taking place. No need to get confused with that particular point. Okay. Now, next question. a very straightforward and a very simple question. Question number 11. Each chromosome during metaphase has two chromatids. Of course, right. And you know very well, DNA synthesis is taking place in which phase of the cell cycle? Undoubtedly in the S phase. Again, some students will get a doubt. Sir, actually DNA synthesis is happening in the S phase, but metaphase means it is the M phase. Sir, then how does the reason will be the correct explanation for this particular assertion? Your question is a very valid question. Anyway, the answer is the A. I want all the students listen. In S phase, what happened, friends? DNA duplication occur. Correct, na? Suppose if they are having a 1C, it will become 2C in the haploid cell. If you take a diploid cell, 2C, it will become the 4C. So the DNA replication started in the S phase. And most important, you have to understand where the chromatin fibers are formed. That is the question. What, friends? Where does the chromatin fibers are formed? Undoubtedly, friends, the chromatin fibers are also formed in the S phase. I hope you're getting my point correctly. But during the metaphase, what is going to happen? They are, I mean to say, during the metaphase, they are having the two chromatid structures. So please don't confuse that there is a no relation here. The point is that the fibers are started forming. The formation of the chromatin fiber occurred in the S phase. So from the S phase only they entered into the G2 followed by the M phase. Then only they can be separated. So the point is that, of course, the reason will be the correct explanation for this particular assertion. No need to confuse with that. Next one. Very simple, straightforward question. Cellulose. Every student commonly know that this is also a kind of intermediate question only. Cellulose. What is the cellulose? Important structural polysaccharide which is present in the plant, which will provide a great strength and stability to the plant, which will provide the mechanical strength. How does the cellulose is made up of? We have studied many number of times. Cellulose is made up to 40,000 of glucoses. Right. So what is the name of the enzyme that is involved in the synthesis of the cellulose? We can call it cellulose synthase enzyme. Right. Now, all the students look into the picture. So cellulose is made up of what? Glucose units only, right? So this is one glucose unit. This is another glucose unit, which is a beta D glucose. When I am explaining you the chemistry in the classes, I will explain you what is this beta position, D position, what is alpha position. You can see that the first sugar, the first carbon of the first sugar associated with the fourth carbon of the second sugar. And they are beta. Beta means, friends, all the OH groups are in the same position. All the OH groups are in the same direction. That is called beta. So beta 1,4 glycosidic linkage. No need to get confused with that particular point. Hence the correct answer is D. Okay. Then the direct simple question. It's not a very difficult question, but important question you have to understand, because EMIS test they use it to ask very commonly in the exam. In EMIS test, if the substance is a mutagen, it will increase the rate at which Salmonella auxotropes revert to true. See, first of all the students has to understand a statement between auxotropic and prototropic. That is a very important statement. What is auxotropic? Auxotropic means which cannot synthesize any substance on their own. What, friends? Which cannot synthesize anything on their own. What is prototropic? Prototropic means which can synthesize everything on their own. That is what exactly we can call prototropic. Now you only look into the picture. Very simple. I have taken the strain of the Salmonella typhimurium. Listen what I have written. It is a histidine negative. Means it is a histidine auxotropic, which cannot synthesize the histidine on their own. which means compulsory it require the histidine on their growth. Now I have taken this and I kept in a growth medium. When I kept in a growth medium, I'm using, so some students again confused. Sir, growth medium means, does it contain all the essential amino acid? No, they don't contain any amino acid. They just contain water, some other micro and macronutrients, but they don't contain any kind of amino acids. Now, how does the histidine can grow? Histidine cannot be synthesized, right? But what I have done, you see this. I have transferred this particular histidine negative medium. Look into this, to the other culture where there is a presence of the histidine. Then what happened? This histidine negative become histidine positive because it has used the histidine which is present here. In other words, I can tell to all the students in a simple words, auxotropic strain converted into prototropic strain. That is what happened here. So what is the question? In MS test, the substance is a mutagen. It will increase the rate at which Salmonella auxotropy can synthesize. What ultimately it is going to synthesize? It is going to synthesize the histidine. Simple, straightforward, direct experimental question. And let me tell you one more important point. What questions we got more in our exam, in our set? All the students keep the point in your mind. Most questions will appear from the ecology chapter. So hence I am requesting all the aspirants. See, it is not like that we should not focus on every other unit. It is mandatory. We have to focus on every single unit, but give the highest importance to the ecology. Such a simple, straightforward, direct question it is. Ecotone, benthic zone, ser and lotic. It's very easy to understand also. First of all, what is lotic? Very simple, friends. There are two terms are there. Lentic, lotic. Lotic means the water is moving in a unidirectional. Continuously water will move, friends. Example, you can take rivers. Correct, na? Where the water is moving in a unidirectional flow. What is correct? What is lentic flow? Lentic means standing water, which means water cannot move. You can take an example of a ponds. You can take an example of a lake. You can take an example of a reservoir. Correct, na? So what's the statement they asked? Lotic. Lotic means running water. Easy to understand. Ser. No need to get confused with this point. Ser means replacement of one community by other. I'll tell you, friends, we all studied in the ecological succession. In the ecological succession, who is the starting species? Every student commonly know that. The starting species is the pioneer species. Yes or no? Of course, yes. Which is the ending species? That also you know very well. Ending species is called as the climax species. Now the question is that between the pioneer and climax, whatever is present, that is what exactly, friends, we can call the ser or the seral community. So the three will be the two. Then benthic. Benthic is a zone between the two different communities. It's a straightforward question, straightforward statement. And what is the no, I'm so sorry. Benthic means what? It is the zone which is found at the bottom of the sea. Suppose this will be the sea, the deeper zone. I'm so sorry. The deeper zone only we generally called as a benthic zone. Sorry. Now, what about this particular ecotone? Ecotone means a zone or a bridge between the two communities. That is what we generally called as ecotone. Hence, what's the correct answer? The answer is number 1. So always you should be very careful, friends, when you are answering the matching questions. Never get confused with that. Next, question number 15. Very straightforward question. In the secretory pathway of protein, today only we have discussed this in the exam. In the class. In the secretory pathway of protein modification, proteins move through various organelles in the following order. So all the students remember my words. if you take a mitochondria, if you take a nucleus, if you take a chloroplast, if you take a peroxisome, so to all these organelles, from where the proteins are coming, that's a straightforward question. For the mitochondria, nucleus, chloroplast, peroxisome, friends, every organelle, the proteins are coming from the cytoplasm. This is an important statement. But friends, if you look into the lysosomes, always remember my words. So endoplasm, from the endoplasmic reticulum, proteins will come to the Golgi complex. From the Golgi complex, they will come to the vesicle. From the vesicle, they will come back finally to the lysosome. Even they will come to the plasma membrane also. So they said chloroplast, peroxisome, lysosome. That is wrong, right? Because they are associated with cytoplasm. This is also wrong because proteins are coming from cytoplasm. Endoplasmic reticulum, Golgi complex, vesicle. This is the straightforward and direct answer. Option D, Golgi complex, mitochondria, nucleus, absolutely wrong because mitochondria and nucleus associated with the cytoplasm. Never get confused with that point. Right, you can see in the diagram very clearly, easy, from the endoplasmic reticulum to the Golgi complex and finally to the vesicle. From the vesicle, it will come to the lysosome. Very simple. Now, another important question, friends. The catalytic efficiency of an enzyme is represented by. First of all, you have to ask me, sir, what exactly the word catalytic efficiency? Efficiency means ability or capability of an enzyme. Ability or capability of an enzyme, or in other words, I can tell you, catalytic efficiency is always talking about the velocity, speed of the enzyme at which the enzyme will combine with the substrate molecule. So this is a substrate molecule. This is an active site of the enzyme. At what rate this particular substrate or this particular enzyme associated with the substrate. So that is explained by Kcat by Km. Right. I want you to always remember this particular concept, friends. In the enzyme kinetics, I have explained it to all of you very elaboratively. It reflects both binding and catalytic events, which indicates how the velocity varies according to how often the enzyme and substrate combine. No need to confuse, very easy. Question number 17. Which immunoglobulin among the following can be transported across the epithelial cell? Always remember, IgA is the antibody which is the dimeric in nature, which is having a two subunits or the two monomeric units. And you know very well, IgA is the secretory antibody which is present in the external secretions. What is that external secretion? Nothing but the epithelial cell. You know very well, IgG is the antibody which is involved in the secondary immune response. You also know very well, IgG is the antibody which cross the placental barrier and provide the immunity to the fetus. You also know very well, IgE involved in the allergic reactions or the inflammation reaction. IgM is the first antibody that expressed on the B cell, which is involved in the primary immune response. Simple point. Hence, the correct answer will be what? Oh, 1 second. What is the question? Right. See, the question they are asking is which can cross the external secretion or the epithelial secretion. So please don't get confused with this point. I think I have mentioned answer as the wrong answer. Undoubtedly, the answer will be IgA. Because friends, please don't get confused. IgG is the antibody which cross the placental barrier. Okay. Whereas IgA is the antibody which is predominant in the external secretions. Please don't get confused with that. Hence, the correct answer is undoubtedly IgA. I think I kept it a wrong answer. Answer is C. Very direct question. Which of the following cell organelles involved in the photorespiration? Every student commonly know that photorespiration is also called as C2 cycle. You know very well, in the C2 cycle, how many organelles are involved? chloroplast is involved, peroxisome is involved, and mitochondria is involved. Remember CPM, chloroplast, peroxisome, mitochondria. So among these three, which they have given, ribosome never involved, lysosome never involved, dictyosome never involved, only the peroxisome is involved. Hence the answer is peroxisome. Very straightforward question. Germplasm preservation through the tissue culture. This is also we have discussed in the question in the classes, which is the ex situ conservation. Correct na friends? What we will do, we will take the genetic materials. We will take the genetic materials. How we can preserve this? By collecting the plant, and we will keep that particular plant in the form of a seeds in the seed bank. By using what? Liquid nitrogen. Again, they will ask this also in the exam. In the germplasm technique, which we use always, which is the liquid nitrogen. So it is we are actually doing the process of preserving, right? Or conserving. So it is not an in situ conservation. In situ means it always happens in the natural habitat. So it is not happening in the natural habitat. It is happening in the seed bank, which is obviously ex situ conservation. No need to get confused with that. You all people studied vernalization. How many times we have studied? Vernalization means what, friends? Vernus. Vernus means what, friends? Spring. That is the meaning. Correct, na? It means spring-like. You know very well. Ultimate goal of the vernalization is what, friends? We have to stimulate the process of flowering. Yes or no? We have to stimulate the process of flowering. How? By exposing the plant to the lower temperature or the cold temperature for a long period of time. That is what exactly we can call vernalization. Increase the ability of a flower. Increase the ability of a flowering by exposing to the long periods of cold winter. Always remember that particular point. Okay. So friends, that's all for now. In the tomorrow class, again, I will continue the part 2. All the questions are very straightforward questions and very direct questions. I am requesting all of you, only if you are really serious about your preparation, please then only call to this particular number or WhatsApp on this particular number for the further information. If you are not very serious about your preparation, I encourage you, please don't waste your time as well as our time also. If you're truly dedicated and you want to crack the exam, then please call on this particular number. Okay. Thank you. Thank you, all of you.